Okay, so welcome to the next video in which we are discussing type 2 activation of endothelial cells. Okay, so we've discussed now many things which happen when you have an endothelial cell which is undergoing type 2 activation. So type 2 activation leads to the activation of activator protein 1 and nuclear factor kappa B. And we've discussed that the activation of these two transcription factors increases the expression of cyclooxygenase 2, which leads to increased production of prostacyclin, which leads to vasodilatation and increased blood flow to the affected area. In addition, we've discussed that these transcription factors increase the expression of unknown effector proteins, which then cause the regression of the cytoskeleton from the edges of the endothelial cells. So they move backwards basically to increase the gap between neighbouring endothelial cells, which increases the leakiness of the capillaries and the venules in the affected area, which leads to the increased production of a inflammatory exudate containing complement and coagulation factors, which then both help in the attack on the invading Staphylococcus aureus. Okay. Finally, we've also discussed how it will lead to the expression of E-selectin and CXC chemokine ligand 8, or interleukin 8, which will both also help with the type 1 activation mechanism for recruiting neutrophils. So the E-selectin will bind to sialyl Lewis X on the neutrophil, and that will tether the neutrophil to the endothelial cell. Then what you'll get is the CXCL, which has been mounted on heparan sulfate proteoglycan on the surface of the endothelial cell, will bind to a CXC chemokine receptor on the surface of the neutrophil, and that forms this tight adhesion. Okay, so you have this juxtacrine signal basically, which contains both the E-selectin and the CXCL8. Okay, then the neutrophil will diapodese through the gap between the two endothelial cells, and we've discussed that that's helped by the PCAM1 protein and also the CD99. Right, however, what we're going to finally discuss is that this happens initially. Okay, so when you initially expose an endothelial cell to interleukin 1 and tumor necrosis factor alpha, these three things will happen: increased vasodilatation, increased leakiness, i.e. increased size of these gaps between the endothelial cells, and increased expression of E-selectin and CXCL8 to recruit neutrophils. However, after around 6 to 24 hours, what will happen is the gene expression will change. So the genes that type 2 activation is activating will change, and the most pronounced change that happens is that you stop producing E-selectin, and instead you start producing two proteins known as ICAM1 and VCAM1. Okay, so let's discuss these two then. So ICAM1 stands for intercellular adhesion molecule 1. Okay, so this stands for intercellular adhesion molecule 1. So this is one of the exceptions, nearly an exception, to the rule that CAM stands for cell adhesion molecule. In this case, the C is within the word intercellular, but it's nearly there. It's so close that it's not really an exception. So the intercellular adhesion molecule 1, and also you're going to increase the expression of VCAM1 on the surface of the endothelial cells. Okay, and this stands for vascular cell adhesion molecule 1. Now, these are going to be involved now in the recruitment of monocytes from the blood rather than neutrophils. So we're stopping with the recruitment of neutrophils. We're stopping the production of E-selectin. So we stop increasing our extravasation of neutrophils. Instead, we're going to turn our attention to recruiting monocytes. Okay, so let me just give a little bit of a discussion as to what a monocyte is. So a monocyte is another form of white blood cell, and these have very large nuclei. Okay, so here is a single nucleus like this, and they circulate within the bloodstream, so this is a monocyte. However, once you have moved a monocyte out of the blood into the interstitial space, it will undergo differentiation and it will become a much bigger cell known as a macrophage. And macrophages, their name means big eater. Macro means big. Phage means eater, so they are a big eater. And they are a very good phagocyte, basically. They will do the same as the neutrophils, but they're more powerful than the neutrophils. They will engulf bacteria into phagosomes. So let me show this. So what will happen is they will engulf the bacterium into a little sort of intracellular vesicle, but it's not what's known as a phagosome. Okay, so here's the nucleus again. So then you'll have the bacteria within this little vesicle that this phagosome that you're creating, so this invagination of the membrane inwards. And this will pinch off, and then what you'll have is this phagosome within the cytoplasm of the cell with the bacterium in there. So let me colour in the bacterium so we can recognise it as Staphylococcus aureus. Okay, so this little vesicle that you've got within the cytoplasm of the cell, this is what's known as a phagosome. Okay, and what's going to happen is that lysosomes, which are vesicles full of lytic enzymes known as lysozymes. Okay, so this is full of lysozymes. Okay, and this is what's known as a lysosome. So this is a lysosome, and it's full of enzymes known as lysozymes. Okay, this lysosome will come and fuse with the phagosome. So it will come over here, fuse with that phagosome, and it will empty its contents. It will empty all of its lysozymes onto the bacterium which is within this phagosome. And these lysozymes will then begin to break down the bacterium. And that's the process known as phagocytosis. Okay, and it leads to the destruction of the cell that has been phagocytosed. Right, so neutrophils do this, so do macrophages. Macrophages are more powerful than the neutrophils. Neutrophils basically are a weaker cell because when you attempt to do this, the way I've shown it is it's a very simple process. You bring the bacterium in and then you just destroy it. However, of course, the bacterium will put up a fight. It will do all sorts of things to try and prevent the phagocytosis from happening. So it doesn't want this lysosome to fuse with the phagosome, and many bacteria have mechanisms for preventing that. And neutrophils are weaker at doing this phagocytosis process than macrophages. Right, so we want to now recruit monocytes from the blood and turn them into macrophages as opposed to neutrophils. So we're bringing in a more powerful cell now. This is like if the neutrophils were pawns, these might be a rook or something along those lines. They're a more powerful player. Okay, right, so we're stopping the expression of the E-selectin on the surface of our endothelial cells, and we're going over to these ICAM1 and VCAM1 molecules. Okay, now in addition, you're also, we need another piece of paper. You're also going to need a molecule for tight adhesion, so it always obeys this juxtacrine signal property. You need one molecule to tether the monocyte, and you then need another interaction for the tight adhesion. Now, both this ICAM1 and VCAM1, they're both really involved in the tethering rather than the tight adhesion. So let me show this. Okay, so here's our endothelial cell here. Okay. Right, and what you do is you put on the surface of this endothelial cell these two molecules, ICAM1 and VCAM1. but you also have another molecule which you put on the surface, which also helps in bringing the monocytes in, basically. Okay, and this other molecule is what's known as CCL2. Okay, so we're going to put another molecule, and again, this is a chemokine which is mounted on the heparan sulfate proteoglycan. So let me put the heparan sulfate proteoglycan here. Okay, right. So here we have this CC chemokine ligand 2. So this is CC, and then it's chemokine, and then ligand 2. Okay, and this would often be abbreviated to CCL2 for short. Okay, so these two molecules here now are our ICAM1 and our VCAM1. Okay, and both of these will have their expression increased in the later phases. Later phases of type 2 activation. Okay, and we'll continue this discussion in the next video.